I'm a senior student studying biotechnology department. I'm so glad to be with you today. I'm going to talking about genome editing topic. Well, genome editing can be defined as any change that happens to a sequence DNA specific in the cell or in the organism. It can be made by specific enzymes called engineered nucleic enzymes. This enzyme is mainly used to add, remove, or alter DNA in the genome. By editing the genome, characteristics of the cell or an organism can be totally changed. Well, did you ask yourself how does genome editing work? Let me explain. At first, genome editing uses type of enzymes called engineered nuclease, which cuts the genome in a specific place. Engineered nuclease made up of two mainly parts. The first part is nucleic part that is responsible for cutting the sequence of specific DNA. The second part is DNA targeting part that is designed to guide the nuclease enzyme to cut its sequence. After cutting, the cell naturally using its enzyme repair to repair the cut caused by engineered nucleases. We can manipulate this repair process to make changes in DNA that locate in the genome. Okay, the types of genome editing are divided into three types. Small DNA changes, and we can remove section of DNA, and we can add a section of DNA. After that, what is genome editing used for? Have you think before? What is the main purposes of application of gene editing? Okay, it's used in many purposes, but the mainly three purposes are for research. We can change the DNA sequence to understand its biology and understand how they work. Number two, to treat diseases. We can modify human blood cell to re-enter or re-put it again in the body to treat some conditions such as leukemia and AIDS, and some conditions as muscular dystrophy and hemophilia. The last one in biotechnology, used in agriculture and used in genetically modified plant in order to use increase its yield and use plant resistant to drought, as well as genetically modified cattle that have no horn. Last but not the least, ethical consideration. There are 2 points in ethical consideration. The first point is the safety. It shouldn't be used for clinical purposes because the rate of harmful is higher than the rate of potential benefit. The second one is informed consent. It's impossible to take an informed consent from the patient because genome editing affects mainly the embryos and next generation, which is not born yet. Finally, it's CRISPR-Cas9. CRISPR-Cas9 is the most common, cheap, and efficient system that is application for genome editing. It's a natural process found in bacteria. The bacterial immune system can defend itself against the infection of bacteriophages and conjugation of foreign bodies that attack these bacteria by its DNA, by destroying and bind to its pathogenic DNA and cut it into two pieces which cannot be bound again because the happened of mutation. This is illustrating video showing that the invasion of bacteriophage inside the bacteria. Here, bacteriophage pour its DNA inside the bacteria, and CRISPR-Cas9 molecule will bind with the bacteriophage DNA. The RNA in the CRISPR-Cas9 bind completely to a section of the bacteriophage DNA, causing it to cut. So this DNA of bacteriophage cannot be bound again cannot be bind again because it have been occurred mutation in it. Thank you for your time.